Good morning. Myself, Dr. Ahmed Abdul Haleem Khan, the coordinator of today's lecture workshop, sponsored and supported by Science Academies on COVID-19 Basics to Clinical. It is my proud privilege to welcome all of you to the two-day lecture workshop, sponsored and supported by Science Academies. I welcome the dignitaries online, Honorable Registrar Professor Naseem Ma'am, Honorable Convener of the workshop, Professor Tarala Nandedkar Madam, Professor B. Vidyavardhini Madam, Dr. Nishigandha Madam, and my colleagues, beloved colleague, Dr. Jalandhar and Dr. Srinivas. Now I request Professor B. Vidyavardhini Madam to give her inaugural address of today's lecture workshop. Over to Vidyavardhini Madam.
Good morning to all the dignitaries who are present, our esteemed Registrar, Professor Naseem Madam, the convener, Dr. Tarala Nandekar Madam, and Dr. Haleem Khan, and everyone, all the resource persons. I, on behalf of Botany Department, welcome you all to the two-day lecture workshop on COVID-19 Basic to Clinical. And it's indeed a privilege to be hosting this, as we have hosted so many lecture workshops, and we are very, very indebted to all the three academies for giving us the opportunity once again to host this. I welcome one and all and thank you one and all. God bless.
Thank you, ma'am. Now it is time. Our Honorable Registrar Ma'am will address the gathering on inaugural session of two-day national workshop. Professor Naseem Ma'am. Ma'am, over to you. Naseem Ma'am.
Thank you very much, Dr. Haleem Khan, sir. Esteemed speakers of Science Academies virtual lecture workshop on COVID-19, Basics to Clinical, organized by the Department of Botany, Telangana University, Dr. Nishigandha Nayak Madam, Director HITRT, Mumbai, Emeritus Scientist Dr. Yogesh Sir from NCC Pune, convener of the event, Dr. Tarala D. Nandedkar Madam, retired Deputy Director, ICMR Mumbai, Dr. Haleem Khan, Dr. Ahmed Abdul Haleem Khan, sir, coordinator of this event, head of the Botany Department, Telangana University, Professor Vinita V. Deshmukh Madam, Sanskrit Samhita Department, distinguished staff of Botany Department of Telangana University, Professor B. Vidyavardhini Madam, co-coordinator of this event, Dr. M. Aruna Madam, delegates and participants. A very good morning to all of you. I am pleased to have the opportunity to be the part of this event. I would like to begin by thanking and congratulating convener and coordinator of this event for hosting and moderating this event in this pandemic situation. It is very important in this present prevailing situation, and it is a great initiative. It is the need of the hour for our self-care to save lives during COVID-19 crisis. In this COVID-19 pandemic situation, as we move forward, we know that the pandemic continues and healthcare community must continue to deliver high quality care to all patients. We can emerge from this emergency only by working together. It is important that as a medical practitioner, doctors, scientists, and other health professionals. As human beings, each one of us should be committed, dedicated in preventing spread of this disease by following precautionary measures of COVID-19 and ensure that our treatment plans for our patients according to the latest evidence. Physicians and healthcare professionals have worked hard. They have risked their own health to serve their patients. They are the great heroes during the challenging time. In the present prevailing situation, yoga should be performed daily at home with family members using only yoga resources, online yoga resources to fight against COVID-19 pandemic, as it helps in boosting immunity, it enhances body's natural defense system, it provides a ray of healthcare as it helps in getting rid of many diseases and leads to long and healthy life. I am sure that this Science Academy's virtual lecture workshop will be an opportunity for all the participants to meet and discuss about recent innovative methods in the concerned field. Deliberations, discussions, and exchange of views would be constructive, useful, and motivating. I hope it will render fruitful results to our healthcare system in this pandemic situation. I wish this lecture workshop a grand success. I wish you all a great future ahead, happy, healthy, and peaceful, long life. Thank you. Thank you for giving me an opportunity.
Thank you, ma'am. Thank you so much. With kind words of Registrar Ma'am, Honorable Registrar Madam, Professor Naseem Ma'am, we conclude the inaugural session, and we will move for the next part. Now, I would like to request Professor B. Vidyavardhini Madam to introduce our today's first resource person of the two-day lecture workshop, Dr. Nishigandha Naik Ma'am, Hafkin Institute for Training, Research and Testing, Mumbai. Please, ma'am, kindly introduce Dr. Naik Ma'am.
Good morning to one and all. Yeah. Good morning to one and all. It gives me immense pleasure to introduce our today's speaker, Dr. Nishigandha Naik Ma'am. Dr. Nishigandha Naik is working as an assistant director and also held an additional charge as the director of the Hafkin Institute of Training, Research and Testing, Mumbai. In her capacity as assistant director, she served as the head of the departments of biochemistry and toxicology. She has more than 30 years of research experience that includes stints in academia as well as industry. Her work in the field of cancer cell biology, imaging, snake venom, and pre-clinical drug discovery. Her research primarily relates to cell mortality-related signal transduction events and development of diagnostic and therapeutic approaches for tuberculosis and cancer, especially chronic myeloid leukemia, breast cancer, glioblastoma, and neural stem cells. Studies of her group have identified raw GTP phases as therapeutic targets. She is also involved in research on snake venom and establishment of the facility for the testing of snake venom and anti-snake venom as well as National Venom Research Center. Dr. Nayak did a PhD at the Center Research Institute, CRI, of the Tata Memorial Institute, which is a pioneering and leading comprehensive cancer center of India. She did her postdoctoral research at the Center of Études Nucléaires de Grenoble, France. She worked as a principal investigator at the CRI, now known as Advanced Center for Training, Research and Education in Cancer, ACTREC. The focus of her group was to understand pathogenesis of chronic myeloid leukemia and breast cancer. In a tenure of almost 10 decades at CRI, she has developed expertise in the field of imaging. She was instrumental in establishing the Institute of Laser Confocal Facility, which was the second kind of its in India. She contributed to the flow of cytometry and live cell imaging. facilities of the institute and served as a member of the academic committee, institutional scientific committee, etc. The NCE research division of the Piramal Enterprises Limited was focusing on the discovery and development of new drugs to treat unmet medical needs in the areas of cancer, diabetes, and inflammation. The center had comprehensive capabilities ranging from a target identification, lead discovery and optimization to preclinical development and clinical research and development. As a senior group leader, Dr. Nayak initiated a work from the establishment of a group and the laboratory too. She undertook tasks from target identification, discovery, and development of lead compound to the preclinical stage. One of the compounds that reached the clinical stage trials. As the head of the imaging cancer biology, she had set up a complete preclinical imaging facility required for the drug discovery industry. Dr. Nayak is an inventor in international patents and is a recipient of several national and international awards, including the Indian National Science Academy Medal for Young Scientists, Award for Young Scientists from the International Federation of Cell Biology, IFCB, and European Cell Biology Organization, ECBO. Marie Curie Fellowship from the Commission of the European Communities and the DST Long-Term Overseas Associateship Award from DBT. STA Fellowship from the Government of Japan and DST, etc. Dr. Nayak has served several times as the elected office bearer of the Indian Association for the Cancer Research and the Indian Society for Cell Biology. She is the founder president of the ACTREC Alumni Association and is associated with the Moving Academy of Medicine and Biomedicine as a member of the Governing Council. So with a vast experience, ma'am, we thank you very much for accepting and we thank you very much for being in the seminar. God bless, and over to ma'am. Haleem Khan.
Thank you very much for your kind words, Professor Vidyavardhini.
Thank you, ma'am.
Can I share my screen?
It's already there, ma'am. Your screen is being shared.
Ma'am, you can.
Okay. Okay, so is my screen visible now?
Yes, yes, it is very much clear.
Okay.
Morning, ma'am.
Good morning. Professor Naseem, Registrar of the Telangana University, Dr. Tarlan Andedkar, convener of the workshop, Professor Ahmed Abdul Haleem Khan, convener of this lecture workshop, my dear colleagues in the university, as well as co-speakers and student friends. It's indeed a great pleasure to be here as a part of this workshop. I'm really thankful for inviting me for this workshop because this gave me opportunity to go a little deep into today's challenge, which is a COVID-19. As you might have heard, this is not really my area of the work, but since all of us are trying to find out what is the latest and how we get really answer to this challenge, this was a good opportunity for me. So not spending much time with the introduction, I would first like to give a brief list of events that has been happening throughout 1 year. The outbreak of unusual pneumonia was first reported on 17th November in China, which was in the province of Hubei. Later on in December, the first official report of the occurrence of this unusual pneumonia from Wuhan city was reported by China. After this, so today you must have realized this is almost 1 year now since the time this unusual pneumonia has been reported. So what was the causative agent of this? Since it was very unusual, people tried to find out what was the cause because there was a fatality which was very much associated. The first case which was identified in November, as well as the first 4 cases which were identified in December led to fatality of the patients. That's why there was a great need to find out what was the causative agent. And what was reported by looking at the genome sequencing, deep genome sequencing, is that the causative agent or the infectious agent belongs to the coronavirus, but it is not one of the known coronavirus, but it was a novel coronavirus. So when it was reported, the first time it was sort of named as 2019 novel coronavirus, 2019 NCOV, COV stands for coronavirus. This was on 12th January. So you can imagine how quickly, because of the technology, the events have been occurring, and we are progressing really very fast. Now you all know we have been listening to the news of the vaccines which are coming up, and it's not just one single vaccine, but a couple of vaccines are coming up, and we are really looking forward to it. Coming back to now what has happened after 12th January, it was later on, that is by end of 30th January, by end of the January, that is on 30th January, I think my slide is not moving properly. Okay, so on 30th January, the first case of COVID was identified in India. And on 11th February, it was named as a severe acute respiratory syndrome coronavirus 2, that is SARS-CoV-2, because there was SARS virus, which was known, and this virus was found to be very close to that. The symptoms were similar, but rather more dangerous. And that's why finally the International Committee on Taxonomy of Viruses labeled it as a taxonomy as a SARS-CoV-2. Later on, the disease has to be identified, and that's the responsibility of WHO. So in international classification of diseases, WHO put this as a coronavirus disease 19, COVID-19. That's how the terminology COVID-19 came into the picture. Now this COVID-19 was officially announced as a pandemic on 11th March 2020. So within 3 months from the time of identification, this pandemic has really occurred, the way it has spread all over the world. Now, when I say it has spread all over the world, how many countries are really having coronavirus or COVID-19 patients? So as of now, there are more than 215 countries in the world which have patients of the coronavirus. I'm giving you a statistic of 2nd December, just because it's kind of a midway between the November event, which was unreported officially, and December, which was officially reported from Wuhan. So more or less at the beginning of December, probably it's 1 year which we have completed. So on 2nd December, as per the WHO statistics, what we see here, that in world, there are 63 million identified cases of corona. And out of this, 1.4 million led to the death of the patients. While in India, there are about 9.5 million cases, and out of these, about 1.3 lakhs are fatal. Now, when this is such a severe thing, actually the question is, what do we do? Since it was similar to SARS-CoV, can those medicines be used? But nothing was really working on this virus. So there was a big hunt how to really resolve this issue. Now, let's first look at the central dogma. I'm just taking to the basic, because I understand there are even undergraduate students in the audience. So please pardon me if I'm going into too basics, but I think this is essential because it's very important to know how any virus functions. So if we understand the central dogma, what happens is from DNA, by transcription, RNA is formed. And if it's only RNA virus, then by reverse transcription, you get DNA, and finally you get RNA. And ultimately for RNA, proteins are formed, which are actually playing role or have a function in any of the organism. So once we understand this central dogma, let's see what was known for coronavirus from the dip sequencing that has occurred or the electron microscopic studies. So coronavirus was found as a very large envelope, positive single-stranded RNA, about 30 kilobases. To be precise, 29903 base pairs were found in coronavirus, and it has a 5' cap structure and 3' poly A tail. This was responsible for infecting not only a variety of human, but now I'm talking in general about corona. So please don't get mixed up with coronaviruses when I say it's a terminology, and when I say SARS-CoV-2, that's specifically for SARS-CoV-2. So coronavirus, basically a huge virus. In fact, this is one of the largest RNA virus, and its diameter is in the range of 60 to 140. And for SARS-CoV-2, this is reported to be around 120 nanometer. The outer surface of the coronavirus has surface spikes or which are about 9 to 12 nanometer long. And these spikes are actually the one which play critical role in infecting the host cell. So if you look at the structure of corona virus, what you see now, this is a structure of SARS-CoV-2, which is published by WHO. So what you see is this gray matter is nothing but the lipid bilayer envelope of the virus. The yellow dots that you see are envelope proteins, while the orange one that you see here are membrane proteins which are denoted, and the red ones are the spike proteins or S protein. So this is a general structure for coronavirus as well as a specific one for SARS-CoV-2. Now coming to the little more in depth. Now, when we say classification or this is classified as a coronavirus, what are the parameters that one generally uses for classifying the virus? As I started with the genetic material, which is now easy to find out these days because of the various technologies available to us, genetic material is the first criteria, whether it's a DNA virus, RNA virus, whether it's DNA or RNA, then whether it's single stranded or double stranded. These are the first thing that lead us to going ahead in inventing any of the infectious agent. For virus, then the next question is whether it's enveloped or not enveloped. What is the size or shape of the virus? Then what is the intracellular location, whether it's nuclear, whether it's cytoplasmic, then which are the hosts for virus? Does it infect human? If it is infecting any human being, then is it coming directly or there are some intermediate hosts? This is very important when it comes to control of the disease. And lastly, but most important, is the mode of spread of infection, how the infection actually occurs. Now, considering all this, let's see now what are the things that are happening. Now, if we look at, excuse me, the genetic material classification of virus on the basis of genetic material and envelope, then what we see here is, sorry, there is some. So what you see here is, if we talk of DNA virus, then it's double-stranded or single-stranded. If it's double-stranded, then again, whether it's enveloped or not enveloped. And these are few examples which are given. For example, enveloped double-stranded DNA viruses are HIPAA-DNA viruses or herpes virus, pox virus, while unenveloped or without enveloped DNA double-stranded viruses are adenoviruses, papilloma viruses, polyoma viruses. While if we think of single-stranded DNA viruses, these are generally unenveloped. The best example of this is a parvovirus. Now coming to RNA viruses, again, whether it's a single-stranded or double-stranded, whether it's a positive strand or negative strand, depending on, there are further classification of the RNA viruses. If it's a positive strand of RNA and it's unenveloped, then examples of these are calciviruses and picoma viruses. While if it's enveloped and positive strand, then this is where the coronaviruses actually come into the picture. Other than that, there are flaviviruses, togaviruses, which are a part of this single-stranded RNA envelope viruses. If it's a negative strand of RNA and it's enveloped, then few examples that I would like to give are arenavirus or fleovirus. While if it's double-stranded RNA viruses, then it would have a capsid. For example, in case of reovirus, while if it's a positive single-stranded RNA, then one of the best example is a retrovirus. So this is how the viruses are classified depending on the genetic material as well as the presence of the envelope on the virus. Now, other than this, shape of the virus is also very important. These are few examples that I have given. It could be a helical structure or a tube-like structure. It could be a polyhedral with protruding spikes or without spikes. It could be enveloped, without envelope, as we have discussed earlier, or it could be a complex structure where you find a head, tail, and then protruding tail so that it can really penetrate into the host cell very well. Now, these are just few examples where you see electron micrograph or X-ray crystallograph constructions of viruses. This is how they actually look. Now, this is a tobacco mosaic virus, the first one which was invented, and we all are familiar from our graduation days. So what you see is a real rod-like structure of the viruses. This is electron micrograph. Well, this is the turnip mosaic yellow virus. Now, what you see here is a spherical shell. This is now not by the electron micrograph, but this is X-ray construction. So there are various technologies by which we can found structure, a shape of the virus as well as the structure of virus. Now, the third example here is a bacteriophage T4. We are again very familiar with this, and this is now a cryo-EM. So in the technologies are different, but what we get is a good resolution, and depending on the virus size, the kind of envelope it has, the technology that one uses differs. Now, just few examples of various shapes of virus. Now you see vaccinia virus, which is just with envelope, while paromavirus have envelope which have slight spikes. Similar is the case with herpes virus, but it has further envelope also along with the membrane. ORF virus, which is a kind of oval structure, rhabdovirus, which is again a rod-like structure, T4, which I told you earlier. Then there could be kind of flexible structure. So the tail of the virus, the way which it was in T4, which was a rigid structure. While in case of flexovus tailed phage, what you see is a long tail, which is very flexible. So it helps virus to probably move into the host cell. Then adenovirus, which is again hexagonal and it has lots of spikes. Influenza virus, or small viruses like polyomavirus, picovirus. So this is just to give you a flavor of different kinds of shapes and the virus which we are familiar with, how they differ from each other just by looking at it. These are again just few structures. And now I'm coming to the coronavirus. Now, if you compare coronavirus with all other viruses, what you see here is you have nice decoration, like a crown. The whole virus is decorated. And that's how actually the family name corona has come into the picture, because it has a crown like a structure on its membrane all around. And that's the indication that it's coronavirus very easily. While if you say another virus like herpes virus, what you see here is a spiky structure and not nice decoration the way you see in case of corona. Other than coronavirus, again, few examples are given which actually show you in the earlier sketches also, like smallpox, rabies virus, hantavirus, hepatitis virus. So now the question that comes into the mind is, why is it important to look at the shape? Why is it important to know the genome structure? See, when we don't know how to treat the virus, then we have to find how the virus enters into the host cell. And if it enters into the host cell, what are the pathways where we can actually stop it going into the cell? Now, if we want to stop something, that means we need to interfere with either the genome or with the structural proteins of the virus so that we can stop at the stage of entry, or when it enters later on with its genetic material, if you can manipulate and stop the replication of the virus. Now, if we look at the coronavirus are broadly grouped into four different genera. Alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. Now, if you look at the gene structure of these, they have a long, at the 5' end, a long chain, which actually gives two major proteins, polyprotein 1A and polyprotein 1B. And this has an identity of about 58% among all these different genera. Well, if you look at the C terminal, then these are majorly structural protein. I'll come to this a little later, but this has identity about 43% among the genera. And overall, if you see, then identity is about 54%. Now, this shows that identity is very little in structural proteins. That is because every virus has different hosts. So how it goes into the host cell is important. If they now what we see here is beta genera is where coronavirus lies. So whatever you are using for other viruses, whether it's, for example, it's Hong Kong virus, which was Delta virus, or Alpha virus. Now, whatever we are using to stop infection by these viruses cannot be used for this because they share very little identity. So the drug that you are using will not be very effective. And that's why one needs to know what is the genetic structure so that we can find out similarity and have our targets chosen accordingly. Now coming back to this polyprotein 1A and polyprotein 1B, that is PPA1A and PPA1B that I'll be referring to. These form non-structural proteins, while the structural proteins that are formed by the C terminal are majorly four proteins. S protein or the spike protein, then envelope protein, membrane protein, and nucleocapsid protein. Now again, if these are few examples here, like for example, alpha coronavirus, what you see is HCOE229E. Now these are depending, the nomenclatures are depending on where they are inverted, what are the similarities. Example of beta coronavirus, where the COVID causative agent lies. What you see something similar is MHV, that is murine herpes virus, SARS-CoV, that is SARS coronavirus, MERS coronavirus. We all are familiar because we have seen what havoc these two viruses created earlier. Now, if you go to gamma coronavirus, it is infectious bronchovirus. Now, if you look at this SARS virus, MERS virus, or bronchovirus, all of them are affecting respiratory system. But what you use for IBV control, you cannot use for SARS or MERS, simply because there is such a vast difference that whatever you're using, the way you're targeting this virus will not work on this. Now, the last one is Delta virus, which is HKU11. Now, HKU11 is Hong Kong University, which recognizes this virus, and that's how they have named it as HKU11. So knowing that there are basically two proteins, non-structural proteins and structural proteins, and identity lies more at the non-structural protein. Homology is more between non-structural protein than structural proteins. Now let's see how to address and how do we derive the structure of 2019 nCoV or SARS-CoV-2. Now this is a detailed phylogenetic relationship in coronavirus family. So Coronavirinae is the subfamily which has 4 different genera, as I have stated earlier. One is the alpha coronavirus. Now beta coronavirus has again 4 different lineages in it. Lineage A and lineage C are very common in Europe and America, while lineage beta, lineage B or beta of the beta coronavirus is seen in East Asia. Lineage D is not really seen all over the world. And again, what you see in the rest of the Europe or in the Middle East area is the mutated of the lineage C that is found and reported. So this now shows how much is the distance between these different viruses which lie in the coronavirus family. And if you can see, which I doubt because it's a bit too small, but the coronavirus 2 is close to the structure of RATG13, which is a bat coronavirus. There is almost 95% similarity between RATG13 and coronavirus SARS-CoV-2. And this is one of the reasons, if you remember the initial news about coronavirus, whether it is coming from bat, because of the similarity that was seen after sequencing, it was one of the prediction which people thought very likely. Now SARS-CoV, which has created have a few years earlier because of the infection. Now, if you look at the similarity, it is little distant than the bat coronavirus. So there is no much similarity. We'll go a little later in depth of the similarity between SARS-CoV and SARS-CoV-2. But what I would like to draw your attention is, this is completely different than the rest of the lineages of the beta coronavirus. Now, how many viruses of these coronavirus are pathogenic? Now, these are few examples which are very important one, which I have quoted here. Now, if you see CoV229E and CoV-NL63, these affect human and they create mild respiratory infection. While now these PRCS, ISU1 or TGEV, PUR46 MAD or PED, I mean, there's no point in going and reading each and everything. But what I would like to say, another species which is affected by coronavirus is pig. Now, in pig, what you find is respiratory infection as well as the digestive tract infection, or digestive tract is affected other than the respiratory system. Now, if you go down, even the dogs are affected, and in this, again, diarrhea is seen. If you remember, there are some sporadic reports where the diarrhea has been seen in the patients which were suffering from CoV-2. Some in camel, this infection is totally asymptomatic. It is also seen in cat, where you see both vasculitis as well as fever. Now again, CoV HKU1, CoV OC43 are infecting human. SARS-CoV, MERS, I'm coming to this. But if you see now in this again, pneumonia, mild respiratory tract infection. Now SARS, severe acute respiratory syndrome. What we have seen is 10% mortality rate in this. While in case of MERS-CoV, that is severe acute respiratory syndrome, which happened in the Middle East. Now here the mortality was almost 37 mortality rate. So this is where was a big challenge. But what is happening now is probably worse than that in the beginning, which is CoV-2. Other than now these 6 important human viruses, again, other animals which could be affected by corona are cow, horse, mouse, whale, chicken, bulbul, sparrow. So almost major species which are on this globe are affected by this coronavirus. Now I'm just focusing on classification of coronavirus which affect primarily mammal. So if you take family Coronaviridae, the subfamilies, as I have shown you earlier, one of them is Coronavirinae and another is Torovirinae. Now we're in Coronavirinae genera. What you find is alpha, beta, gamma, and delta virus, which we saw in the phylogenetic structure. In alpha coronavirus, now what I have a color code here. Those which are in green are those which affect human, and those which are in red, they affect human very badly. I mean, you have a great, rather they affect badly and they affect through ACE2 receptor. So HOV229, which is from alpha corona, and HCOV NL63, both these affect human being, but HCOV NL63 is the one which is going to the host cell through ACE2 receptor. In beta coronavirus, again, HCOV, OC43 and HKU1, they are affecting human being. While in beta lineage B, SARS-CoV and SARS-CoV-2. Now, if you look at, if you remember rather earlier phylogenetic classification that we have seen, in this family, these are close in beta lineage, and the bat one is slightly different because the primary infection is in the bird and not in the human. But structure-wise, again, SARS-CoV-2 is close to bat CoV. But entry of bat CoV is not through ACE2 because it doesn't have any color code. But if you see the lineage C, another one which is important CoV is MERS-CoV. And the entry of this is again not through ACE2. So probably it's not the entry that matters a lot, whether it becomes fatal or not, but the structure and how we actually use this structure to derive therapeutic molecule. Then again, lineage D, one of the examples that affects also human is CoV HKU9. Now knowing this, and ACE2 is one of the host receptor where coronavirus bind, let's see how this CoV2 goes. But before that, as I had mentioned earlier, one should also classify the virus depending on its origin and its transmission. Then when you don't have any medicine, at least you can control the transmission and try to stop the spread of the virus. So let's go one by one. Now HCOV OC43. This affects human host, but the natural host is rat and intermediate host is cow. And this has actually occurred in mid-1960s. And again, I have used a color code here. These yellow arrows show that this doesn't give very severe infection, but the infection is comparatively mild and could be controlled. Now, the next coronavirus which came again was in 1960, which was HCOV229E. Now, here the primary host was bat and intermediate were animal, especially deer, but it did affect human and gave respiratory syndrome. The third one which came up was in 2003. I'll come to this later because they are categorized as a separate which are highly infectious. So the third one, which is in the mild one, is HCOV NL63. Now this was in 2004. Again coming through bat. We don't know what is the intermediate host here, but it does affect human. And the fourth one, which gives mild infection, is again HCOV HKU1, which is through rat. Now this was in 2005. So if you see, these are one after another. This has come little off late. Now, one thing I would like to point out here, when it's an RNA virus, it has comparatively higher mutation rate because there is no repair system anyway in the virus, and being in RNA, they really mutate much faster. Coming to now those which are severe infection, the first one that has happened or that has occurred, the first outbreak was of the SARS acute respiratory syndrome, that is SARS-CoV. Now here, again, if you see for all the acute infection, the primary or the natural host is bat. Now SARS-CoV, it was a bat, and then it went into the animals and finally come to humans. Now this was outbreak in 2003, while comparatively recent outbreak was of the MERS, that is Middle East Respiratory Syndrome, MERS-CoV in 2012. And being in the Middle East, probably from bat, it had gone to the intermediate host camel, and then it has come to the human. And now what we have is SARS-CoV, SARS severe acute respiratory syndrome coronavirus 2. Again, the natural host is supposed to be bat, and through animals, it has come to human. So knowing this, now we have, so when we didn't know much about the structure, or we are to begin understanding of the structure, the best way to see to stop the spreading of the infection is to control the host, whether it's a natural host or intermediate host. Now this is something which was at the level of the pathogenesis, but how actually the virus looks like. I had explained you that it has a kind of corona around it. So if you look at the structure of the virus, this is a cartoon of SARS-CoV-2. So what you see here is spike protein, which is protruding out, which is about 9 to 12 nanometer in size. Then we have a membrane structure. This membrane is made up of majorly glycoprotein. Also, there is an envelope, which is known as a protein E, and then RNA, which has nucleocapsid protein. So this is overall structure of the SARS-CoV-2. This is the structure of spike protein, which is very important to us because through spike protein, the virus enters or attaches to the host cell and this gets entry into the cell. So if you look at the structure of this spike protein, what you have is a heterotrimer of the S1 subunit. Now these are globular hairs, and on the top of this, we have a receptor binding motif. Followed by this is a S2 unit, which is a stop fusion domain, and this holds, or rather takes hold onto the membrane of the virus. And then this part of it is an intracellular tail. So this is how the whole, it's like a claw which is pierced into the membrane. We'll come to the structure of this little later further. But before that, we'll see what other information that we have got from the genome sequencing of SARS-CoV-2. So if we look at the genome of SARS-CoV-2, what we find is, as I told you earlier, it's about 30K base pairs. At 5' end, you have UTR regions. Similarly, at 3' end, you have UTR region. These UTR regions are followed by ORF1AB. Now, ORF1A and ORF1B gives us PP1, that is polyprotein 1A, and polyprotein 1B. Now, these polyproteins ultimately give you this, if you can see here, non-structural proteins 1 to 4 and 6 to 10. After that, there are some other enzymes like helicase. I'll come in a little later with the details of this. Then we have hexonucleases, non-structural protein 15, ribose methyltransferase, couple of enzymes. This also have enzymes like proteinases, and then it also forms RNA-dependent RNA polymerase. So this is the unit which is very important, though it is not a structural protein. So this is important for replication of the virus. And which are the structural proteins? So this ORF1AB is followed by the protein S. This was followed by open reading frame 3A. Now coronavirus have minimum 6 open reading frames. And this is a structure of SARS-CoV-2, which has 10 open reading frames, as you can see here. Then this open reading frame 3A is followed by E proteins, M protein, then couple of more open reading frames, then nucleocapsid protein, again open reading frame, and 3' UTR. So this is a structure of SARS-CoV-2. Now how it reflects into the functioning of the virus. Now after this ACE is expanded here, which is subunit 1, subunit 2, but we'll address this later. First, let's understand how this genome actually gives us non-structural proteins, since these are very important for the replication of the viral genome. Now, as you can see here, this whole thing is expanded. This region is ORF1A and this is ORF1B, followed by this S region, which is a structural protein. So what you see here in ORF1A, 2 enzymes are transcribed from this region. Papain-like proteases, which is known as a PL2-PRO or NSP3, that is non-structural protein 3. Other than this, there is 3CL protease, which is also known as NSP5. Now, once this sequence are known, structural biologists could derive the structure of these proteins by synthesizing it in the lab and also by crystallography. There are a couple of structures. But what we have found from ORF1A is with these two enzymes, the PP1A, which is formed from ORF1, is cleaved further. Now PL2 or NSP3 cleaves between NSP1 and 2, NSP2 and 3, and NSP3 and 4. Thus, it gives you NSP1, 2, 3, 4, as I've shown you in the earlier slide. While 3CL protease, also known as NSP5, this cleaves at a site LQ-SAG, and this gives a couple of other proteins starting from NSP4 to NSP16. Now, this open reading frame gives you two peptides, as I told you, PP1A and PP1B. Now, these are formed because there is a shift of the reading frame. So just from this small sequence, it's a big sequence for the virus, but this gives rise to 16 by change in the reading frame. We get two proteins. Now, ORF1B has a very unique enzyme, which is RNA-dependent RNA polymerase. This is actually a complex. So this ribosomal frame shift, as I said, happens here, which gives rise to enzyme RDRP. Now this RDRP is actually a formation of complex between NSP12, which we have got here. Then it combines with NSP7, NSP8. It also combines to NSP13 and 14 and form a unit, which is RNA-dependent RNA polymerase. Now, NSP13 is nothing but helicase, and ORF1B also have endoribonuclease, which is a very peculiar thing for SARS-CoV-2. I'll come to structures or function of these all NSPs little later, but before we go, just a quick look at the S protein. What you see here is spike protein, then open reading frame, which are here, are shown by gray regions, then envelope protein, membrane protein, and nucleocapsid. And here is a spike protein. A structure is shown by structural biologists. We'll come to the functioning of this later in the slides. Now let's see what are the functions of this non-structural protein coronavirus. Now this is very important that we should know structure functions of these NSPs, because unless until these functions are not known, we don't know which protein to target while targeting our therapy. So if you look at NSP1, it is involved in cellular mRNA degradation and inhibition of interferon signaling. NSP2, the function is not known. While NSP3, it cleaves polypeptides, block host innate immune response, and promotes cytokine expression. I'm sure you all remember the cytokine burst that happens. So it is very important to see how these proteins function. Now NSP4, it helps in double membrane virus double membrane formation, while NSP5, as I said, it is involved in IFN signaling, polypeptide cleavage, which we saw earlier, and then it also helps in synthesis of proteases. NSP6 is restricting autophagosome expansion and DMV formation. NSP7 is a cofactor with NSP8 and NSP12, as we have seen in RBRD, that is RNA-dependent RNA polymerase, which is a very important and a crucial protein structure in COVID-2. Then NS8, again a part of RNA-dependent RNA polymerase. NSP9 is involved in dimerization and also in RNA binding. NSP10 is a scaffold protein for NSP14 and NSP16. Function of NSP11 is still not known. NSP12, primer dependent of RDRP, that is RNA-dependent RNA polymerase. NSP13 is helicase, which is a 5' triphosphatase. NSP14 is something which is a unique to COVID-19, that is SARS-CoV-2, which is an exoribonuclease, and it has N7 MTAs. The exact role is not known, but it is very unique. So being unique, it could be one of the good target for therapy for the COVID-19. NSP15 is endoribonuclease, and evasion of double-stranded RNA sensors is possible because of this. And NSP16, the last protein of the non-structural protein, it negatively regulates innate immunity. It is avoiding MDF5 recognition, and that's how it regulates the innate immunity. So these proteins are important in functioning of the or formation of the viral replicon. Now coming to this RDRP, that is RNA dependent RNA polymerase of SARS-CoV-2. Now if you look at the structure of the NSP12, which is the major protein in this, what you have are subunits in this: neuron, then there is an interface, and there is a protein which is again further divided as a finger region, which is G and F, then a palm region and thumb region. So if you see here, this is a top view of that protein structure of the protein. So this green thing that you see is NSP 12 fingers. The pink is a thumb, and this purple is a palm of this protein. Now what you see in brown color here and here, these are NSP2 molecules which bind with this NSP12. And this black region that you see is nothing but NSP7. So this altogether forms a complex. Now this is a front view of that which you can see. So you can see a groove here. And then there is again hairpin helicase which is going in. There is an interface protein. So this whole complex actually interacts with RNA. So it holds RNA here. Once RNA is inserted, what you can see here very well. Now this complex of RDRP complex and RNA, here the thumb protein and the palm, that is pink and violet, they give actually support to this RNA and hold RNA. So the RNA could be inserted very well. Now you can see this here, that this is holding the RNA strand and it gradually slides on it. Now this is very important to know, because unless until we don't know how these proteins behave, how they interact with each other, we cannot really find solution. And that's why the structure of the protein is very important. Because anytime if somebody asks you, what do you know about structure? What is the use of it? Then use is we can really control by knowing the structure. Just by knowing once, and now these are all non-structural proteins. So even if we don't know the structure, they're part of the RNA. So when I say structure, it's in a broad term, whatever is involved in the formation of this virus SARS-CoV-2. So once we know how this RDRP really functions, we can go ahead for the structural part, which is forming the unit of virus. So as I mentioned earlier, S protein. Now this is a spike protein which was protruding as 9 to 12 nanometer spikes on or forming a corona of the SARS-CoV-2. Now this is divided into two units, S1 subunit and S2 subunit. So S1 subunit, as if you could remember, this S1 subunit is a claw-like structure which has three regions, and it has N-terminal domain and C-terminal domain. Sorry. And what is sitting in between is domain that binds to the receptor. Now, as you all know, this binds to ACE2 receptor. This RBD, that is region binding to the receptor of the host. And what you see in the middle of that is the motif that binds to the host receptor. So this is very crucial part of the S1 subunit. So imagine there were three subunits of S1. So these three subunits go and sit onto the ACE so that they have a very strong hold on the receptor of the host cell. Now this S2 subunit, which has a fusion peptide, and the other regions involved are the heptide repeats HR1 and HR2. So they help the S protein to actually get itself embedded into the viral protein. So this is a good bridge between the host cells and the virus. So once this, let's see now how this actually goes and sits onto the host cell. So before that, quickly, let me summarize what are the function of different proteins other than S protein. So as I mentioned, S protein is a homotrimer of S protein, makes up spikes or viral surface. It has multiple functions. It has S1 heads, which recognize a cognate cell surface receptor, and the trimeric membrane anchored unit, which contains fusion machinery. M protein, now this is the most abundant protein and has three transmembrane domains which bind to N and E, that is protein N is nucleocapsid and protein E is envelope protein. So they actually determine shape of the virion, promote membrane curvature and viral assembly when the replication of virus takes place into the host cells. Then the E protein, now this is the smallest protein in viral membrane and localizes in the ER as well as Golgi of the host. So this helps actually to have a communication and form a membrane of the virus, and this naturally facilitates virus-like peptide formation along with the membrane protein and nucleocapsid. And the fourth and the last one is the nuclear protein or N protein, which contains two domains which bind to virus RNA. This is agonist to interferon and viral encoded repressors of RNA interface. Now this binds to NSP3 to help tether the genome of RTC and packages encapsulated genome into virions. So now we know what are the functions of the structural protein. We know what are the functions of the non-structural proteins. So let's see now how the whole thing functions. Now this is a kind of cartoon which tells you how the virus enters into the cell. So what you see here is a virus structure, the spikes, then various proteins, NSP1, NSP5, ORB3, ORB6. These are the spikes. These are ACE2 receptors which are present on the host cell. So now why is SARS-CoV-2 goes into the many cells? Because many cells have this ACE2, which is protruding on its cell membrane. So naturally it has a good hold and it could attach itself to the ACE2 receptors and enter into the cells. Now once it enters into the cell, it can just go inside like a trap, or it can form a fusion kind of thing and enter into the cell. Now, why we are trying to look at this then? Because we could know how we can really stop entry of the virus into the cell or replication of virus into the cell. Now, different approaches we are used for therapy, you're going to go in detail of that, but just to give you a brief flavor of it. Now, if you have recombinant SSE2, which can trap this, so the entry could be inhibited. Now, once virus goes in, the genomic RNA starts going to start replication. Now, this could be inhibited. Now, we have heard about remdesivir. Now, how the remdesivir actually stops it, we'll come to the interaction of remdesivir. But not only remdesivir, there are other similar proteins also like favipiravir and all, which function in the similar matter. They stop actually replication of virus. So that's why the remdesivir was tried at a cell culture level. In the lab it worked, but unfortunately it didn't give really promising result when it was used in the clinic. Now, so this is another step where one can control. Then the entry itself could also be blocked by using chloroquine and hydrochloroquine, which was initial use in India to a great scale, so that host immunity is modulated, and that inhibits actually entry of the virus into the cell. Now, once the virus replicates, the RNA is replicated. Now, in the Golgi, the proteins are gone, and then they are sort of packaged together, forming a new virus. At the same time, since it has a binding capacity to various cytokines, it goes and gives immunity boost. Now, this could be now stopped because if you use inhibitors like tocilizumab, which binds to IL-6 or IL-6 receptor signaling, then this whole cytokine burst could be controlled to some extent. So for this, it is so. If you look at this diagram, this part is actually dealing with the entry, and this part is dealing with the signaling that happens because of the virus. So this is just a brief, to give you a brief idea how it could work. Now let's see what happens with its entry, or how the viral protein binds to ACE2 receptor little in depth. Now what you see here is protein S. 2 protein S molecules, 3 protein S molecules here, as you can see. These are the receptor binding domain, 3 domains of the 3 units that are very clearly seen. Now, this is just further shown like S1 and S2 unit. So this receptor binding domain is seen. Then if you recall, there is a central C-terminal domain, N-terminal domain. So once we know this structure of the virus, now what you see here is, this is actually ACE2, that is the receptor for this. And what you see here is this RBD and RBM. RBD, if you remember, is a receptor binding domain, which is in blue, which is in this green color, blue color, and receptor binding domain is in the green color. So to this ACE2 receptor, this receptor binding motifs, they go and bind here. So if you can control here, you can control actually entry of the virus. So this is how there are different approaches which we can use to control entry of the virus into the host cell, or control its replication, or to control the immune response that is given by the host, so that what happens is regulated well and you get the desired immune response than having the cytokine burst, which is sometimes not really in favor of patient's health. So I hope with this now you would be convinced that knowledge of structure of virus will enable us to understand the pathophysiology of the virus and also to explore much needed treatment modalities, especially in case of virus like SARS-CoV-2, with SARS-CoV-2 in case of COVID-19. So I think that's all from me. And if you have any questions and if time permits, I'll be happy to take that. Thank you very much. Over to you, Dr. Haleem Khan. Hello, can you hear me?
Before he says anything, I'm really thankful to you, Nishigandha. You made it so simple, but it is so complicated thing. So it was really very nice listening to you. I was off and on on the video, but I was listening to you throughout. It was really wonderful presentation, right from A to the end. See, to Z, I must say. You've done a
Thank
very
you
good
very
job.
much.
Thank you so much. And you've taken a very nice presentation, and it was very clear also. Thank you so much. Let me pass on to these people. They can.
Yeah, I think I'm not able to come out of this, share my screen. So if organizers could help me in that.
If you close yours, then they can talk, I think. I think so.
Ma'am, good afternoon.
Yeah, okay, fine.
Okay. You want me to mute?
No. Ma'am, good afternoon. A very big thank you for accepting our invitation and giving such a wonderful talk on the detailing of structure of virus. And it is an immense pleasure on behalf of the entire Department of Botany and the Telangana University. I wholeheartedly thank you for giving time and opportunity to know the detailing of the coronavirus in a correct time, because as now the second round of the corona is going to commence, so hence it is once again we thank you.
It's my
Thank you
pleasure.
very much, ma'am. Now it is time, as there is a small change in the schedule. Now actually Dr. Yogesh sir is now in his meeting. So we see that the next lecture by myself will be made now. Instead of Yogesh sir will join us in the post-lunch session. So kindly regret for not his availability. Thank you, ma'am, once again. And now
please excuse me, I'll have to go for another commitment.
Sure.
It was really nice to be with you and helpful for me to understand more of the COVID-19.
Okay.
Thank you.
Thank you, ma'am. Now I would start my talk, my lecture on novel coronavirus. Good morning, respected convener of today's workshop, Dr. Tarla ma'am, and Professor Vidyavardhini ma'am, Professor Aruna ma'am, and the respected participants of today's workshop. It is my time to give a briefing on the current happenings around the world and in India, especially. So I will begin with the concept that pandemics. In medical terminology, there are terms like epidemic, sporadic, pandemic. Like this time, pandemic is a common term, and this pandemic is not a very new. So the pandemics are quite common to the human to the mankind. So we can see that these pandemics begin from ADs like Antonine plague is there, then Justinian plague is there, then Black Death is there, then smallpox was there, Italian plague was there, plague in London was there, and similarly, the plague was the most important pandemic which causes millions of death in the era when medicine or the field of medicine was not developed. It was in a developing stage. So soon followed by that, a great flu was found in 1889 to 1890, and followed millions of people died in that flu. And then followed by that, we have seen yellow fever, and then the flu has continued in the various forms like Asian flu, Hong Kong flu, swine flu, like so many flus were there which killed millions of people. So this flu is a form of viral problem which has been very common in the various parts of the world, and it spread very fast, no time, to the whole globe. Like swine flu of recent times, Ebola virus, then Zika virus, then is SARS. Just now what we heard, SARS, first time in 2002 and 2003, and MERS, Middle East Respiratory Syndrome, and in middle there was a problem like AIDS, which was because of HIV. So in the total history of pandemics, we can see that the deaths are more common due to the viral pathogens to humans, and the host for this is primarily humans. Along with humans, the hosts were intermediate, like sometimes birds, sometimes livestock animals, like many. And in middle, there was only one major problem. We can see like AIDS. It was a typical problem because of blood transfusion which emerged, and then it soon found the various modes of particularly transmission. Like this, the story of the concept begins with the Antonian plague, it was a pandemic broke out in the Roman Empire and passed to various Roman Empire cities, then Italy, Greece, etc., reached Spain, Egypt, and North Africa, and killed more than 2,000 people per a day, and it lasted for long time. And then Justinian plague, which was in 541 to 542, the source of the disease was found rodents and the fleas, which killed more than 30 to 50 million humans. And likewise, there we can see the Black Death. It was a typical kind of pandemic found in two continents, that is Europe and Asia, and near about it killed 25 million people alone in Europe. The Black Death had symptoms of fever, chills, and we can see that the swellings were found all over the body, which were having blood and pus, and found late in the times that viral problem was the more predominant in the form of smallpox. And in Asia and America, we found that 5 lakh people died per annum due to this major problem. And then found in 1871 to 1824, 1 million deaths for the again a major problem, that is cholera. The cholera is a common problem because of bacteria, that is Vibrio cholera, and it was first emerged in India 1817, and it was a common problem because of food and water contaminated by the bacteria, pathogenic bacteria Vibrio cholerae, and it reached to India through the British troops who were traveling to India from other countries of Asia. So this problem was not origin in India, but because of the Britishers who traveled from other parts of the continent to India were the major source of this pandemic in the country. And then followed, we can see uh, Russian flu. It was in 1889 to 90. And it was estimated that 1 million people died because of Russian flu. And it started in St. Petersburg, and slowly it started spreading in the world, various parts of the world. And then followed the story of today's corona, that is Spanish flu. Spanish flu is the deadliest in the history, and it infected one-third world population and killed near about approximately 20 to 50 million people worldwide. There were 3 waves of flu found in the form of common flu. It hit in the spring, and second wave fall in the same year was more deadlier than the first wave, and it killed within hours or a few days after the onset of symptoms. And the third wave came following the year, was deadly and added more death toll. So overall, Spanish flu was the first episode in the pandemic history which created major damage to the mankind. And in 1968, it was first time it was found that H3N2 pandemic, which is a flu pandemic in 1968 because of influenza virus. This virus was commonly first located in the Southeast Asia. Within 2 weeks, it emerged in Hong Kong in July 1968. By December, the virus has reached United States, United States of America, United Kingdom, and other countries of Europe. It killed an estimated 1 million population of the world. and followed by the flu. There was again major viral problem in the history of pandemics, that is human immunodeficiency virus, which is causing a common disease, acquired immunodeficiency syndrome. Though 65 million people were infected with this virus, and it was a common disease because of the sexual transmission. Already the treatment and there was no particular vaccine available, but the disease has come to the control by using more and more precautionary measures. And in 2002 to 2004, we can see the first episode of severe acute respiratory syndrome that was reported again in China in February 2003. It started in China earlier in the November 2002. After a few months, it moved to North America, South America, Europe, and Asia. But it infected less number of people and killed more or less people. So with this, it was an concept that viral problems are more self-limiting after the peak or after reaching a particular peak stage of symptoms. More commonly, it will come down. So SARS was a typical kind of pneumonia kind of symptoms which was affecting the respiratory system. This is the actual concept. What made is more important to be known. The more important thing to be known here is a picture. This picture is a public record of meteoroid strike and can be found at a space.com website, which is available from October 13, 2019. In the website, it was given a title that Brilliant Midnight Fireball Lights Up Sky over Northeast China. It was the event found in the October, and it was told that there was a strike of meteorite. Meteorite was found. People said that current coronavirus epidemic in China is having an event that is called Bright Fireball event that occurred on 11th of October over the Songjia city in Jilin province of northeast China. It is important that this place, Songjia, is hundreds of kilometers far from Hubei, but people say that this episode is having relation with particularly the Hubei episode of the coronavirus. It is told in the report that a fragment of this, whatever meteorite is there, it was happened to be with a carbonaceous material, and it was told that trillions of virus or bacteria were found in the particularly the dust what was falling in the area that entered mesosphere stratosphere with a speed of near about 30 kilometers per second, and people told that it was an envelope carrying biological cargo and dispersed. So it reached mesosphere, stratosphere, and then troposphere. So this fireball struck 2000 kilometers north of Wuhan, and it deposited the particularly, it is expected that some viral particles might have fallen in the particularly area, and people feel that whatever the origin is there, origin of coronavirus is somewhere related with particularly cosmic kind of event. People feel that the emergence of the coronavirus is a cosmic. So these reports were recently published in a book chapter by Edward Steele et al. And they say that this episode of virus, whatever the virus is there, it is a cosmic concept. And there are number of explanations given by the authors of this concept, that this coronavirus is interestingly has come from space in the form of the meteorite. So though it is a concept uh, told, but John Hopkins University has totally nullified this concept that the whatever coronavirus is there, coronavirus is a not a cargo come from meteoroid, but still this is a very interesting concept that whatever coronavirus is there, the coronavirus has its origin from cosmic range or cosmic level was interesting concept what has been explained. Followed by this, as already you have come to the nomenclature, how coronavirus has been named, it is given over here. So the surface of the virus has the particularly appearance of a crown-like structure. So it was given the name as particularly corona, crown from corona. So human coronavirus were first identified in mid of 1960s and named based on its appearance in the electron microscope. And this virus is a diverse group of virus that infect humans and other animals. So here are the various forms of common group belonging to the coronavirus. So this were found in various livestock like and other wild animals like bat, and livestock like pig and poultry and sheep and cow, camel, like so many animals are there which are the potential host, or we can say mammals and aves. Mammals and aves are specially affected with this zoonotic infection. So as per Indian Express, it is told that coronavirus is a group of virus. It is zoonotic in nature and transmitted between animals and people. And human, the virus is known to cause respiratory infection from mild, and it may become lethal. And the other forms, as discussed by the earlier speaker, the three different forms are there, like alpha and beta are there. In this alpha we have two forms and beta two forms. 229E NL63 are alpha forms of coronavirus, and OC43 and Hong Kong University 1 is a beta virus. So the present virus is a beta kind of virus, and other beta viruses as Middle East, then severe acute are the common beta forms. Origin. It has been originated in Wuhan, where the seafood was sold in the market illegally. It was found that the animals were sold illegally, and they told that too many animals were sold here, but majorly snakes were sold in a raw form in this market. So Wuhan virus, did snakes play a part, is a common concept discussed by a medical journal of medical virology has told that the scientists compared the new virus with 276 virus genomes that infect bats and snakes, and they found that it was almost similar to the virus found with the snakes, and they think that it could be formed by recombination, the result of mixing of viruses from different species. And the genetic recombination changed a protein which may allow the virus to enter the human cells. So initially, this was the concept in the early days, told that whatever the coronavirus, novel coronavirus is there, the novel coronavirus has some relation with snake, and felt that the wild animal sellers have sold the snakes, and the snakes were the cause for the spread of virus. And this is the market from where it was told that this was the place from where it was told that this is the location in the Wuhan of Dubai where the animals are sold. In the tubs, you can see that whatever the animals are there, like frogs, fish, and the other whatever animals are there, the other animals are sold raw form, and by wearing nylon gloves, the man used to cut the animals, though he himself is safe. But the whatever people feel that whatever the vessels they use, the vessels used were dirty and the surrounding areas were dirty, and it is expected that among these animals, whatever the animals are there, these animals could be the one among these animals what they have slaughtered, one among the animals may be found to be infected with some kind of coronavirus, and it soon started spreading in the flesh of the other animals which have been slaughtered and mixed up. Though the person is not affected, but the other people who consume this flesh raw were the source it is expected. And this is the turtles put in the vessels with the cage, and then also you can see the flesh of the snakes which have been sold openly in the market. And in when we talk about the COVID-19 especially, though in medical journal of medical virology, it is told that snake could be having the very near relation with this concept. The people told in the early days that whatever the December 2019 China is there, the natural host was horseshoe type of bat, and then the intermediate was also there, that is pangolins, which have been given in the picture. And deaths were approximately at that time in the December, it was the fatality rate was 3.3%, and the source from National Health Commission is told that 2,619 people were died in that out of the 79,359 people. And soon after that, it was declared that unexplained pneumonia has occurred in Wuhan city of Hubei province. There is a lockdown has been declared, and stay measures were taken to stay safe. And soon it was found that the disease from Hubei has reached to various parts like Beijing and Hong Kong and so on. It has started slowly spreading. So in the initial outbreak, it was found that in the different dates from 31st of December to the January 2020, 20th January 2020, we found that the disease was slowly spreading from one place to the another place. In China itself, the death toll was very low. Like in Wuhan, only out of 198 people who got infected, 3 died. And in the other places like Beijing, Shenzhen, and other places, other countries, soon from there, the people, when they migrated from Wuhan, they found that in Thailand, Japan, and South Korea, the disease was slowly spreading. And the Chinese authorities have declared, identified the virus as a novel coronavirus by January 7th of January. And by 12th of January in 2020, WHO has found it as to be the emergency coronavirus infection as the emergency. And soon from there, we can see the picture, satellite picture is showing that how the disease has been moving from one place to the another place, infecting the people, humans. So it is told, as it is very common now at this moment, that this disease is moving by airborne transmission. It is a particularly infection by sneezing or cough. The sneeze or cough of a person releases droplets, and then if the droplets are inhaled by a normal person, then the infection soon enters in this normal person from a diseased person. And soon after that, it was found that the virus was spreading through close contact by touching or shaking, making shake hands. and touching the object or surface with virus is also one of the major consequence of spreading of the virus. And they have told that in the initial days it was the incubation period was 14 days, and soon after the incubation period, fever, cough, and shortness of breath was the major thing. How does the coronavirus affect the body? The coronavirus affect the body majorly. It is having the effect by we can see that the fever symptoms are fever, headache, running nose, cough, and then we can see that soon the other organs will get affected, like respiratory system followed by kidney. In the people who are having it is a secondary disease of death cause, and it is found that in 80% of the people the disease was particularly a very mild version, but in people who are having other disease other diseases like cancer, diabetes, this disease was very, I mean to say, panic. It was very problematic. The 5% of the people who are having the critical illness had respiratory failure, septic shock, and multi-organ failure. And then the treatment. When we talk about the treatment, it was a major concept has come in a recent time, that the BCG vaccine. The BCG vaccine is it was very much discussed that people who got the vaccine called BCG is a vaccine given to prevent the it was found that the BCG vaccine given in childhood, generally in the infancy, is they have found that in the early days of the coronavirus infection, it was told that people who got vaccinated by BCG vaccine, which is containing Mycobacterium bovis attenuated strain, were less affected. They are less affected with the coronavirus infection. It was found that whatever the BCG vaccine is there, BCG vaccine usually improved innate and adaptive immunity by forming various interferons, tumor necrosis factors, etc. And it improved the immunity, though the research is going on in various parts, like different countries like Netherlands, Germany, UK, Australia, and people wanted to root out whether really BCG vaccine is supporting the it is reducing the burden of coronavirus infection. Likewise, it is still a topic of debate, but it was found that in the world countries which got BCG vaccination are less affected, or death toll was less in these places where BCG vaccination was given in the childhood or infancy. So this was the major thing which is discussed. And then the treatment by using the, as discussed by the earlier speaker, it was found that the chloroquine forms, it may be a hydroxychloroquine or chloroquine. It is found that this is an anti-malarial drug. So BCG vaccine is a vaccine of tuberculosis, and it was found that this vaccine, countries where this vaccine was given, were less affected with the corona infection. One concept. And the second concept, that anti-malarial drug administered in the patients of SARS-CoV-2 are more, they recovered very fast, and like chloroquine was earlier given to malarial parasite infection, and also in some other infections, systemic lupus erythematosus and rheumatoid arthritis and some anti-inflammatory diseases. It was found as to be an immunomodulator, and majorly they found that in a COVID-19 infection, there was a huge increase in the cytokine concentration in plasma, and they've told that whatever the hydroxychloroquine is there, hydroxychloroquine reduces the anti-inflammatory response. That is, it reduces the cytokine, and decrease in cytokine concentration in after administration of hydroxychloroquine could be one of the reason that cytokine population decreases, and the complications what further are there could be calmed down. So like this, the BCG vaccinated people are less affected, and people treated with hydroxychloroquine are getting soon recovery from the infection are the two common concepts. And followed by this, there are several approaches coming. There are several approaches coming by the different scientific agencies, like the Moderna Inc. Has made RNA-based vaccine, and then Oxford and AstraZeneca is almost ready to give the vaccine. And in Germany, we can see the Pfizer company. In China, Sinovac has come, and Novax is also in US is making the vaccine, and trials are going on in the primary healthcare people. And in British also, some tobacco firm has come for making the vaccine, and Glenmark is also trying to make the trial of the uh, drug like favipiravir and umifenovir. It is the drug which is tested in the patients of India to reduce the burden or treat the disease. And now I will talk about the particularly the recent, to say yesterday, whatever the newspaper has given the headlines. Then yesterday's newspaper headlines were the people over 14 crore are tested for the particularly till date. Till date, we can see that the people tested are 14.8 crore people were tested for COVID-19, and now they have found that by Monday, today is Wednesday, so by Monday it was found that the fresh cases were less, and the as less cases have come now, the test could be come down now. And it was found that again, this is from NDTV update. It is told that the daily COVID cases in 5 months tally crosses 97 lakh. So 97 lakh people have come, and approximately the people who died due to this pandemic are 1,40,958. And the vaccine has the vaccine has come with particularly the US regulations by Pfizer, and BioNTech vaccine is going to come. And the recoveries, as per the Union Ministry of Health and Family Welfare on Tuesday, they have told that new active cases are 3,83,866, and recoveries are 39,045 fresh recoveries. And this is worldometer, which is saying around the globe what is happening. Around the globe, we can see that the coronavirus, whatever coronavirus cases are there, the coronavirus cases are given in the digits, and whatever the 68 million people are affected, and deaths are particularly 1,054,000 554,402 people died, and recovery is 47 millions 1,90,369 are there. And overall, this is the country-wise case distribution which is shown in this picture. And the high case, when we look the world meter about the COVID-19, when we check out the COVID-19 cases, this we find that whatever the cases are there, 22.59% cases are from United States. United States is standing first in the COVID-19 severity. In the when we see the number of people infected in the particularly United States of America, generally the 22% is the highest infection rate. So we find that whatever the number is there, the number is very high. It is particularly the 22.9% is the particularly the number 32.59% is the particularly the percentage of infection in the United States of America. Then followed by that, we can see the second place. The second place today is the particularly the infected cases, though the deaths are less, but it is found in the volumeter that the 14.27% in India, and followed by that in Brazil 9.7% are their case distribution, and then we find 3.69% in Russia, and France is 3.37%, and Italy it is 2.56%, and United Kingdom it is 2.55%, and we can see the last in this picture is Spain with 2.52%, with the different cases in millions and lakhs of people getting infected. And now we can see the death, especially we have to focus on the deaths. We can see the United States again stands top in the death. We can see the 2,90,443 people died because of the disease. And in India, it was told that 1,40,994 people died because of the infection. And Brazil also, we can see 1,77,388 people died. And Russia, we can see it was 43,597. And in France, we can see 55,521 people died. And in Italy, 60,606. And United Kingdom, we can see it is 61,434. And Spain, it is 46,646 people died. And Argentina, in South Africa also, this disease was very, very effective in and killing more number of people compared with other countries. So 39,888 people died. In Colombia, we can see 37,995 people died. In Germany, 19,539. And Mexico, it is 1,10,074 people died because of coronavirus. And Poland, we can see the deaths are 20,000, and Iran 50,000, and Peru it is 36,000. So the death cases, when we check the death cases with number of days, whatever the number of days, they have made a graph of 250 days versus the patient count, how many patients were there. Based on that, this graph says, again, the topmost, whatever the graph is there, the graph is reaching, when we check the world meter graphical presentation, we can see that again the top region, the top place is for United States, and the least place we can see over there is particularly the Iran. Iran was a place which has less number of cumulative cases in this 250 days. And then they have compared the both countries number of days versus patient count. When they have did the comparison graph, they found that Europe was more affected than America in the number of cases in the different parts of the countries. And now this is a graph, and this is a picture from the Hindu newspaper English Daily, and it says the numbers of the confirmed cases, and also the deaths, and total recovered, and the active cases presently in the particularly country. It is shown, and they have compared this from the 30th of January to November 22nd of November. So they have found that in the map of the country, when you see the map of the country, the map of the country is very clear with different colors, and we can see the different colors. When the color is very dark, it is the complexion of the color says more severity is found. So here are the places where Uttar Pradesh, Maharashtra, Tamil Nadu, and Karnataka are Kerala, or the places These are the places where, and Andhra Pradesh. So these are the places. The severity is more dense color is shown in this picture in the map, where Maharashtra is found more number of cases and more number of deaths the entire country. And next we can see the states like Uttar Pradesh, then the Assam, then we can see the Karnataka, then Andhra Pradesh, Tamil Nadu, and Kerala. Equally, the death toll was equal in these places, and Maharashtra stood top first. And compared with this, we can see that the death toll was more severe in particularly the Maharashtra. And now, when we talk about this pandemic begin in country, it was begun in 30. The three infection cases occurred for the first time in India. People who come on 30th January and 3rd February, in between the 30th January and 3rd February in Kerala, people from Wuhan, China, were came to India, and within a month later, 3rd March, two more cases were reported. One patient had traveled history from Italy, while the other in Hyderabad visited Dubai. So people from Wuhan, people who traveled from Italy and Dubai. In Kerala, people from China were the source of infection, and in uh, Hyderabad, the person from Dubai was the reason for the disease to get. And then soon they found that a person in Jaipur also came from other country, and likewise it was slowly entered the, and by the 15th of April it was found that the travel visa was restricted, as the pandemic was on its peak for its spread, morbidity, mortality of the disease. And on 22nd of March, Honorable Prime Minister of our country has declared a Janta curfew of 14 hours. And soon after that, we can see that on 14th of March, first phase of 21 days lockdown was started. Due to this lockdown, various things like uh, mm, uh, stations of visits, parks, workplaces, and recreation, pharmacy, groceries, except grocery and pharmacy, all the locations were closed. And the government declared second phase of lockdown on 3rd of May, and it lasted till 17th, and later it was extended to 31st of May. So lockdown and social distancing was very effective, and the Quarantine Law of Epidemic Disease Act 1897 was implemented, and the 123-year-old legislation allowed the country to inspect people traveling by railways, ship, airways, and segregate suspects in hospitals under temporary accommodations. Soon after the first case in 30th January, the total number of patients reached 107 by 15th of March. Since then, the number of positive cases increased, and within 15 days, the confirmed cases were 10 times multiplied in the country. And by 30th March, the 1071 cases were there, out of which 29 died. And ICMR was projected to reduce the cases by 62% by social distancing and the interventions of quarantine. So in this total scenario of pandemic, ICMR, that is Indian Council of Medical Research, has worked anonymously and made the country to decrease the pandemic situation. And uh, it has suggested the government to increase the testing, and stringent measures to be taken, and implement the different restrictions. And the ICMR has played key role a key role in controlling the spread, through lockdown and social distancing. And major cities, some of the cities in the country were the hotspots of COVID-19, like Ahmedabad, Bengaluru, Bhopal, Chennai, Delhi, Hyderabad, Indore, Jaipur, and Kolkata. These were the these are the cities in the country, metropolitan cities, which accounted 40% of the COVID cases in the country. And the disease was very high in elder people who have the other health issues. By 11th of July, COVID-19 cases were 2 lakh 29 lakhs 2 lakhs 92,258 cases, and the people who died, the recovery rate was 60.86%. So like this, the present Ministry of Health and Family Welfare has given the numbers last night, and these are the numbers which have been found in the website of the ministry, where it was found that the Andhra Pradesh was with 5,626 cases, and presently active cases are there. And next is the Assam with 3,585. In Bihar, it is 5,467. And then again in Chhattisgarh, we can see 19,589 active cases are there. In Delhi alone itself, we can see 22,486. And Gujarat, 14,493. And Haryana, it is 12,000. And Himachal Pradesh, 7,895. Jammu Kashmir, 5,000. Jharkhand, it is less. Again, in Kerala, we can see 59,607 active cases. Madhya Pradesh, we can see 13,443. In Maharashtra, again, the toll is very high. Still, we can see the peak number of cases. The peak number of cases are there in Maharashtra. Again, 76,852 as per the records of ministry. We can see the total active cases in the country present till tonight. Yesterday tonight, it was 38,38,066 are the active cases in the country. So with this data, what I pulled to give the global scenario, I come to an end of my presentation. So over to Tarana madam. Tarana madam, please can you speak? Tarana madam. Ma'am, are you able to listen me, ma'am?
Yes, yes.
Please, ma'am, your response, kindly you give your...
Yes, definitely. Definitely, I will talk to you. It's a very nice presentation. Especially, you didn't just talked about the coronavirus, but also the other viruses or other pandemics which were earlier. The world has seen. Some of them India has seen, but you have given a very good review of everything, pandemics as well as about the corona, the global scenario. Thank you very much. Thank you. You want the questions to be asked later on, or how is the situation? Because if there's some time, maybe if students want to ask questions, they can be allowed to ask questions if it is okay for the. Can you carry on?
Vidya ma'am.
I think everybody seems to be hungry. I think your mic is not on.
Oh, yeah. Good afternoon, everyone. Good afternoon, Tarla ma'am. And participants, if you have any questions, you can ask any questions to Dr. Haleem Khan. We'll have a 10-minute interaction so that you can, before we break up for lunch. You can unmute yourself and
Aruna ma'am, can you review the both presentations, what you have seen? Good afternoon, ma'am.
Good afternoon, sir.
Yeah, can you quick review both of us, whatever presentations are there, whatever we presented?
Yes. So good afternoon to all the participants who are attending this Science Academic Virtual Lecture Workshop for 2 days on COVID-19, basics to clinics. And today was the first day. And we had inaugural by our Registrar Madam, Professor Naseem Madam. Then we had 2 lectures. One lecture was by Professor Nishigandha Nayak Madam. And she has given a very good talk on structure of novel coronavirus, SARS and COVID-2. And we have learned a lot and we are thankful to Madam. And next, it was Dr. Haleem Khan sir, who is head department of our botany, Telangana University. And sir has narrated all the history of emerging viruses right from the period of 18th century to the present day. And it was a real, lively presentation, and hope that all the participants and hopefully all the students might have learned a lot. And we are thankful to the science academies who have granted and sanctioned financial assistance, with the support of whom we are in a position to organize today's virtual lecture workshop, where we are having participants from all over the state and from all over the nation. And hopefully we are awaiting for the next 2 speakers to deliver the talk. And I think it's a time for lunch, and we'll be back after lunch. Over to you, sir. Thank you.
Thank you, ma'am. Thank you very much.
Can you tell us what time we are expected to come back?
1:15.
Okay.
Dr. Yogesh sir will be joining us by the time post lunch. So kindly join us again back in the afternoon session, and please, all participants, try to post your questions in chat box, either in the live streaming of YouTube or in the Zoom itself. You can post the questions, and we will be communicating your questions to the respective resource persons and get the queries to be answered. So questioning is more important, and I expect my students who are joining in the Zoom can lively chat, or lively you can have a chance. We can make you to interact with the resource persons. Like morning, you have got an opportunity to meet a wonderful lady with lot of experience, and you found that the women are especially shining like anything in the field of science. You can see our convener madam, and also Nishigandha Naik madam, and in afternoon post lunch we have Dr. Vinita madam. So these ladies are really the thing of joy, because they enjoy the science. They worked like anything, and they are the celebrities of the science in their fields. So tomorrow you will be able to listen to our convener ma'am, and you can see how women are working and how the women are creating wonders in the field of science. I ever found such wonderful ladies who are working hard and proving the science as an important role in the society. Science as what science can do. So today's disease, or today whatever we are discussing, this disease can be overcome, or it can be the burden of this disease or the death toll can be reduced by the again hard findings of this kind of people. So with these few words, I request, and I take permission of our convener ma'am, so that we will be again joining at 1:15 in the meeting to have the two lectures by Dr. Yogesh sir. He's emeritus scientist from National Center for Cell Science, Pune, and Dr. Professor Vinita V. Deshmukh madam from Ayurved Mahavidyalay, Pune, BSDT, Ayurved Mahavidyalay, Pune, and both the people, Dr. Yogesh sir will be explaining you how the corona or COVID-19 detection, what are the tests performed, for the detection of corona disease. And also Dr. Vinita madam will be giving you the Ayurvedic perspective and experience of her own on cancer patients under COVID-19 pandemic. So we are waiting for two more lectures, informative lectures, which are there in the post lunch. So dear participants, I request one and all to join back again at 1:15 in the same, with the same link. With the same link you can join, and have the two informative lectures again back. Thank you one and all. We'll join again at 1:15. Thank you.